Good evening and welcome to all our alumni, students and staff, and also guests that are joining in for our webinar today. So before I start off, I'd like to acknowledge the traditional owners of the land and pay respects to the elders, past and present. And also if there are any members who are from an Aboriginal or an Indigenous background, I acknowledge and welcome you to this webinar as well. So for those who are joining us for the first time for this webinar session, we run these webinars to highlight some of the research that we do in the various schools within this Faculty of Science. So it's in the benefit of our alumni in terms of ongoing learning, and hopefully the research that we do is helpful for your work and also your research that you may be doing currently. So today's webinar is presented by the School of Biology, and we'll get to the session in a little bit. A little bit of housekeeping in terms of the format of the session today. Look, if you have any questions, please do add it to the Q&A section, which is just underneath your video screen. There's a 30-minute presentation and then followed by a Q&A session. During the presentation, you don't have the opportunity to ask verbal questions, just so in the interest of the time, because it's late in the night in Australian time. So, but please do add your questions into the Q&A as the questions arises. Okay, so I'd like to now move on to the formal part of the session and introduce you to the Head of School of Biological Sciences, Professor Craig White. Just to he'll give a brief update of what's happening within the School of Biology, and then move on to Jeremy and moderate the session afterwards. So a bit of background about Craig. Craig White's an evolutionary physiologist. His research is focused on describing and understanding causes and consequences of physiological variations in animals. So Craig grew up in South Australia, and where he obtained his PhD from University of Adelaide. And then he moved on to working in University of Birmingham for 3 years before taking a lectureship at the University of Queensland. He then moved to Monash in late 2015. He was then appointed the Associate Dean of Research in the Faculty of Science from 2018 to about 2020, and then appointed as the Head of School of Biological Sciences. So Craig's received the President's Medal for Animal Biology from the Society of Experimental Biology and future fellowships from the Australian Research Council. So I'd like to invite Craig to start off the session. Over to you, Craig.
Thanks, Udi. Thanks, Udi, and thanks everyone for joining us. This is one of the more joyous things that I get to do as part of my job. So I might tell you a little bit about biological sciences. There's about 300 of us all up, including honours students and PhD students and our staff in about 35 research groups. Our research is really broad across biological sciences. So we have some of the university and the country's best biomedical researchers, one of whom we'll be talking to today. And we have all the way from biomedicine through to theoretical evolutionary biology and ecology and conservation biology. So to give you a little bit of a taste of what's happening in the school at the moment, I thought I'd tell you about some articles that have been in the news recently. So Francine Marquez, her work's been highlighted, demonstrating the importance of dietary fiber for lowering blood pressure and reducing the risk of cardiovascular events. Paul Sunnix and Sasha Pavlova, their work was featured in a New York Times story about genetic rescue. This is the introducing new genetic variation into populations that are threatened. The work was highlighted about the helmeted honeyeaters, which in 1989, just 50 of those were remaining. And from the same species, they've introduced some genetic variation from Gippsland honeyeaters, and that's helped the population to increase. And it looks like they're doing quite well, but birds are reasonably long-lived, so they're continuing to be monitored. And Anna Peters, the Deputy Head of School of Biological Sciences, her research was highlighted in the press, where she's demonstrated that there's a significant increase in cooperative behaviour and a decrease in aggressive territorial behaviour for fairy wrens during the non-breeding season when conditions are much rougher. So when the conditions get tough, the groups come together to help each other. And that's kind of a nice little relevance to the School of Biological Sciences, where I became head of school during COVID in 2020. It was a rough time for everyone, and I was really struck by how much everyone from the school helped each other out during that time. And the last thing that I'll highlight is an example of one of our great people who's doing great things at the moment is Professor Bob Wong, who was previously a deputy head of school here. He was recently awarded the Australian Council for Graduate Research Award for Excellence in Research Supervision. And it was highlighted by our Vice-Chancellor just earlier today that he is not only one of Australia's best PhD supervisors, he's also one of Australia's best researchers. And it's wonderful to work with people like this. One of the many wonderful people I get to work with is Jeremy, who'll be talking to you today. So he's going to be talking about an emerging treatment for bacterial superbug infections that are no longer responding to antibiotics. And this is through the use of viruses called phages. So he's going to talk about his research and how they're developing novel phage-based solutions to combat some of the most challenging and severe bacterial infections. Jeremy joined the School of Biological Sciences here at Monash in 2016, not long after I arrived. And in 2020, he joined the Centre to Impact Antimicrobial Resistance, and he now leads the Monash Phage Foundry, which sits just below me, where I'm sitting at the moment on the first floor of one of our buildings. So prior to coming to Monash, he worked in a postdoctoral position with Professor Forrest Rauer in San Diego State University, and there he was involved in world-first phage therapy case treating a patient with a disseminated multi-drug resistant infection. His research group at Monash studies bacteriophages and their interactions with bacterial hosts in the human body. He set up the Monash Phage Foundry, which is a manufacturing facility for bacteriophages here in Clayton, School of Biological Sciences. It's the only one in Victoria and one of just two in Australia. His research is getting recognition through funding, media, awarding bodies, and it was recently highlighted in an SBS documentary called Last Chance to Save a Life, and we'll provide the link for that at the end. And just recently, it was announced that he was awarded a prestigious National Health and Medical Research Council leadership level investigator fellowship. And this is in recognition of the translational work using phage therapy to combat antimicrobial resistance. It's wonderful to have the chance to introduce Jeremy because he's not only tremendously successful and insightful, he's also a really good guy to work with. So Jeremy, with that, I will hand over to you.
Thank you, Craig. Really appreciate the introduction. Thank you, Udi, for organizing this. And finally, thank you for everyone logging on. I realize this is a Thursday night, at least for people based in Australia. So I appreciate your time. So I'll make a start. My name is Jeremy Barr, as Craig mentioned. I'm an associate professor at Monash University in the School of Biological Sciences. And I'm going to tell you today about our work looking at using phages to combat antibiotic-resistant bacterial pathogens. And so I want to start really simply, and I'm going to start with this question of what is a bacteriophage? And so really simply, a bacteriophage, or a phage for short, is a virus, but it's a specialized virus that can only infect and kill bacteria. And so being a virus, these phages are actually completely dependent on their own bacterial host for their own growth and replication. They can't replicate outside of a bacterial host cell. They're also the most abundant and diverse organism on the planet. So there's more phages than everything else on the planet combined. So they're incredibly abundant, incredibly diverse, and in my opinion, very, very beautiful. And so they've got these amazing capsids and shapes and structures and different diversity and morphologies. And so I want to take you through the phage life cycle, how a phage replicates. And I'm going to come back and touch on this in the talk a few times. And so this image here is an incredible image. It's from the 1970s, and it's a transmission electron microscopy, and it's a slice through an E. coli bacterial cell. And this image shows 3 out of the 4 major steps in the phage life cycle. And so this life cycle starts with the phage, or the virion, latching onto the bacterial host cell, and it uses its tail fibers, these leg-like structures, to recognize a receptor and irreversibly bind or absorb to the bacterial host cell. Immediately after this, the phage then uses its tail structure to penetrate the bacterial cell wall and cell membrane, and then it ejects its nucleic acid genome inside the bacterial host cell. Immediately as this happens, the phage takes over the bacterial cell. It shuts down all bacterial processes. It stops bacterial metabolism. It starts replicating its own nucleic acid genome. It starts transcribing and translating phage structural proteins. And these form fully infectious phage particles inside the bacterial host cytoplasm. And the final step in this life cycle that's not pictured here is the explosive lysis or death of that bacterial cell, where the phage produces a few enzymes to attack the cell from within, releasing hundreds of new phages back into the surrounding environment to start this process all over again. But the most amazing part about this life cycle, in my opinion, is its speed and its efficiency. And on average, this whole life cycle takes just 30 minutes and can produce up to 100 new virions for each cell infected. So these are really, really efficient micromolecular machines that kill bacteria very, very quickly. So with that broad background into what a phage is, I want to take you back. And I'm going to take you back to 2015. And at this time, I was right at the end of my postdoc in San Diego. And I was really fortunate to be involved in a world first phage therapy case study. And this was a world first in Western medicine. So I'll acknowledge that phage has been around for over 100 years, and they're still used to this day in parts of Eastern Europe. But this was a world first approach in Western medicine where we used a personalized approach to treat a patient who was suffering a life-threatening antibiotic-resistant infection. And this case has become known as the Tom Patterson case, and it's been quite famous and well-known in medical literature. So, like all good stories, I'm going to introduce you to the bad guy first. And so the bad guy in this story is a bacterium known as Acinetobacter baumannii. It's a bit of a mouthful, but this is a really problematic bacterium. It's actually top of the WHO and the CDC's list of urgent pathogens that we need novel antimicrobials for. It's estimated that in 2019 alone, this A. baumannii bacterium was responsible for nearly 500,000 deaths worldwide. It causes significant morbidity and mortality. It's a really diverse pathogen with extensive antibiotic or antimicrobial resistance determinants. So it's a big problem, particularly in hospitals and ICU settings. And so I'm going to introduce you to the patient in this story. And this is Tom Patterson, pictured here. And Tom and his wife, Stephanie Strathdy, are big history buffs. And the start of this story is that they were traveling in Egypt. It was their kind of dream holiday. They'd always wanted to go to see the pyramids. And unfortunately, some point in that trip, Tom started to get sick. He started to develop a bit of abdominal pain and cramping. He thought it was food poisoning, tried to sleep it off. But the next morning, things got a lot worse. And it got so bad that he eventually was medevaced to a local EMT clinic in Luxor, Egypt. And it was there that they diagnosed him with pancreatitis, so an inflammation in his pancreas. This was a really severe condition. So he's medevaced to an ICU clinic in Frankfurt in Germany. And while there, they did an endoscopy and they found that his pancreatitis was actually caused by a multi-drug resistant Acinetobacter baumannii infection, one of the worst bacterium you could be infected with. And so Tom was really sick. He went into ICU. At some point during his treatment, he was medevaced from Frankfurt back to San Diego to the UC Thornton Hospital where Tom and Stephanie lived. And he was there for many months battling this life-threatening antibiotic-resistant infection. So there was some hope that he was going to recover from this. At that stage, the infection was localized in this pancreatic pseudocyst. But at some point during that case, a drain placed in his abdomen slipped, and what was a localized infection spilled out into his abdomen and very quickly became systemic. It got into his bloodstream. He experienced septic shock and started shaking. And it was at that point that it was really clear that antibiotics were not going to save him. And he was facing a very grim reality. And thankfully for Tom, his wife, Stephanie, she recognized this. She recognized and saw what was going to happen. And so she started an international search for alternative treatments. And she came across phage therapy. And so Stephanie led this international search and actually pulled together many, many phages from all across the world to treat Tom. And this is specifically where I got involved in the case. So this is actually a picture of me holding 5 of the phages that we treated and we cleaned up in our lab in San Diego. So we had 48 hours to clean these phages to make sure that they were safe to administer to Tom. And then we passed them back to the medical team who moved forward with phage therapy. And this was enabled through a world-first experimental investigational new drug application that we submitted to the FDA. That was the leading governing body in the US for all food and drug administrations. And they gave us approval to attempt this experimental therapy to try to save Tom's life. So pictured here is Chip Schooley, the lead physician in this case, who led this personalized phage therapy treatment to try to save Tom and fight this superbug infection. And so phage therapy began on the 16th of March in 2015, and the team started with intracavitary administration, so directly into Tom's abdomen. This was given 3 times daily. This was tolerated quite well, but Tom was very, very sick. So the team stepped this up, and 2 days later, they began intravenous administration of those phages directly into Tom's bloodstream through a PICC line, and this was given 4 times daily. And so I want to run you through this clinical case. And so we'll go through this graph here. So on the bottom here, we've got day of illness, and you can see that this is starting at day 90. So Tom had already been sick and battling this for about 3 months before this graph starting. Below, you can see all of the antibiotics he was on, really severe last resort antibiotics. On the left-hand y-axis, we've got his leukocyte count, or his white blood cells. So the higher this is, the higher the sign of inflammation and infection. And on the right-hand y-axis, we've got his maximal daily temperature. And this is in Fahrenheit, and I apologize, you can blame the Americans, but 98 degrees is about 37 degrees body temperature. So anything above that is showing fever, infection, and inflammation. Now, on day 110, which was the 16th of March, intracavitary phage therapy was started. And as I mentioned, 2 days later, on day 112, intravenous phage therapy was started. That night, after IV phage therapy was started, Tom woke up from a coma that he'd been in for months. He spoke to his wife and daughter, and the medical team was astounded. I remember getting an update. That was a Friday afternoon or Friday evening. We got the update that Tom woke up from the coma, and the medical team was astounded. No one had seen a patient this sick recover this quickly following this treatment. So it was a great day. Everyone was quite excited going into the weekend. Unfortunately, 2 days later, on a Sunday, things took a turn for the worse. Tom's blood pressure dropped. His white blood cell count went through the roof. His daily temperature spiked. He went into a fever. His mental status declined, and he slipped back into a coma. And the medical team was terrified. They thought they had overdosed him with phages and that his body was having an immune reaction to these phages. They stopped phage therapy. They put him on broad spectrum antibiotics. I was told it was really touch and go that night. There was multiple times where Tom nearly passed away, but thankfully he pulled through and he stabilized the next morning. And so blood cultures were ordered, and they came back the next day. And they came back showing the growth of anaerobic gram-negative rods, which were not the Acinetobacter baumannii. And so what had happened here is that the phages were so effective that they really reduced his bacterial infection that he woke up. He felt better. He came out of this coma, but he was still incredibly sick. He had multiple tubes and wounds in his abdomen, and he actually had a secondary bacterial infection that was caused by a Bacteroides theta otomicron. This is a gut commensal organism. And thankfully, this was antibiotic sensitive. And so this was the bacterium that nearly killed him, but those broad spectrum antibiotics got that under control, and he slowly recovered. The team restarted phage therapy, and he was on phage therapy for another 4 weeks. His treatment was complex. He had a couple additional bouts of septicemia, but he generally improved over that time period. So post-treatment was complex, but his condition got better, and after 3 months post-phage therapy, he was finally discharged home from UCSD hospital after a nearly 9-month battle with this multi-drug resistant superbug. And I can say he's still alive, happy and well today. And Tom and Stephanie actually wrote a book about this. It's called The Perfect Predator. I have no involvement in this book, but it is an incredible story. So if you wanted to learn more, I'd encourage you to look that one up. But one of the things that really struck me about this case, and that I really took with me moving forward in my career, was that we knew close to nothing about these phages. In fact, the only thing we knew about these phages was that they killed Tom's isolate in a test tube, and we administered billions of these into his body. And so at the end of this case, I finished my postdoc, and I moved to Monash University, the School of Biological Sciences, where I'm still there today, running a research lab looking at the use of phage therapy. And so I'm going to tell you about a story, again, looking at Acinetobacter baumannii and trying to understand the phage biology to leverage that to see if we can apply this and save more patients. And I got to give a shout out to Fernando Gordillo, He's actually our first PhD student in the lab. He's now working at the Alfred Hospital, still in the space, but I'm going to present on some of his work. And so I want to put up this slide here. So this is a slide of our clinical library. So we've got many Acinetobacter strains at the top and many phages on the left-hand side. And an orange square shows that phage can infect and kill that host. And the point here is that these phages are really specific. Typically, they only infect and kill one strain. And so I want to go through how do we find a phage? How do we get these phages that infect these hosts? And the answer is that we fish for them. Apologies for the pun. But instead of using a bait or a regular bait, our bait that we use is a superbug. So we use our superbug and we cast our superbug into a proverbial river or lake or stream, but instead of using that, we actually cast it into the most abundant microbial source on the planet, which is beautiful, amazing sewage, wastewater. And so we use this wastewater as a source to find these phages. So how do you fish in 4 easy steps? The first step is you pick your superbug of interest. We grow these on the lab, we grow these on the plates, and we choose the superbug that we want to find a phage for. Step 2 is you select your favorite sewage sample. I've actually got many. I don't think that's weird. We vortex, we mix the sewage sample up, we centrifuge it to pull out all the particulates, and what we're left with is a supernatant containing billions and billions of phages. We then mix that bacterium with those sewage samples and we grow those overnight in big flasks. And the hope is that one of those phages in the sewage samples will infect and kill that bacteria and will grow up in large numbers. And the next day we mix these and we put these on these agar plates, these top agar plates, and we look for these zones of lysis. And that's a single phage that's able to infect and kill that bacterial host. And so I'm going to tell you about this phage here. This is phage FGO2. It's got this beautiful myoviridae structure. And these little dots here are this phage killing the bacterial host on the plate. So the bacteria is growing in this sort of opaque color. And that black dot there is bacterial death. And so we can grow these bacteria. So what I'm showing you here is a growth curve. So we've got time on the hours and optical density. So this is a measure of how much bacterial growth is present. And over time, we see the bacteria grow and they get to really high numbers and concentrations. Now we can grow this bacteria in the presence of a clinically relevant concentration of imipenem. This is a last resort antibiotic. And you can see it doesn't really affect, it slows its growth, but doesn't really affect its maximal capacity. But when we grow this in the presence of phage FGO2, flatlined, dead, right? Superbug beat. Until we look at the end of the growth curve and we see the emergence of phage resistance. And this is one of the biological phenomena that we wanted to study. So we took these isolates and we studied these phage-resistant mutants to identify the bacterial receptor. What's the phage recognizing to facilitate that infection? So again, here's the wild-type host growing in the presence of the phage in red, and you can see the bacterial killing, and then these phage-resistant mutants emerge. And so we picked one of these, we isolated it, and we're growing that in blue. So this blue isolate is phage-resistant, and it's growing in the presence of that FGO2 phage, and it doesn't care. It's completely invisible to that phage. And so we started to characterize these phage-resistant mutants. And so what I'm showing you on the top here is a scanning electron microscopy image here of the wild-type host, and you can see it's producing this gloopy capsule. It's got these really scary appendages and fimbriae coming off the bacterial cell. And if we compare that to the phage-resistant mutant, it's actually, quite cute and harmless. Little bacterium sitting on the slide there. And below that, we've got a capsule stain. And this capsule stain, you'll see the white halo rings around these bacterial cells in pink. That's the capsule that the bacteria is producing. And you can see that's largely absent in the phage-resistant mutant. So we already had a smoking gun that these phages are recognizing this capsule. And so we wanted to do the genetics. So we extracted the DNA from these bacteria. We sent them off for sequencing. And then we did a comparison. We compared the wild type to the phage-resistant mutant. We found a single mutation in the gene known as GPI6. This stands for a glucose-6 phosphate isomerase. I'd never heard of this gene before when I first saw it. And what we saw was a single nucleotide deletion at nucleotide position 823 in this gene, which led to an early stop codon, which interrupts that gene and makes it non-functional. And this gene was present in the capsule locus, a group of genes responsible for the production of capsule. And so we started to identify this gene as the receptor for that phage. And so we generated a knockout mutant. We took a kanamycin resistance gene, which is one of the few antibiotics this bacteria was sensitive to. We put it inside this GPI6 gene, and we used homologous recombination to knock that gene out. And we then did a test to show that these phages could not infect this mutant. And so what I'm showing you here is an absorption assay, and we're measuring, or we're looking at the loss of free phage over about a 30-minute period. So I'll walk you through this graph. So here we've got the wild type, and you can see the wild type bacterium is absorbing about 1 to 2 logs of phage. So it's absorbing those phage because the phages are infecting it. The phage-resistant mutant in dark blue doesn't absorb any phages. It's completely immune and invisible to those phages. When we knock this gene out in light blue, we can see we recapitulate this loss of phage absorption. And when we put that gene back into the knockout on a plasmid, we can restore phage infectivity, thereby confirming that, yes, the capsule is the receptor for this phage. But that capsule is really important. It's a very important structure in the bacterial cell. And so losing the capsule allows the bacterium to be phage resistant, but this came at a cost. And so we found that these capsule deficient phage resistant mutants were highly sensitive to killing by the complement system or human serums. This is a natural immunity that's in our bodies. It had expanded sensitivity to additional phages. And perhaps most excitingly, we found that these capsule deficient mutants were resensitized to antibiotics they used to resist, and particularly beta-lactam antibiotics. And this ceftazidime or ceftaz, this changed from a clinically resistant to sensitive phenotype. And so, bit of a recap here. So these phage-resistant mutants, these phages use the bacterial capsule as their receptor to infect the bacterium. This led to a emergence of phage-resistant isolates that had loss-of-function mutations in capsule production genes. But that capsule is a really important structure in the bacterial cell. And so as a result, these capsule-deficient phage-resistant mutants were susceptible to serums, additional phages, and additional antibiotics. And so what we're proposing here is this sort of concept of phage therapy 2.0, where phage therapy 1.0 was what we did in the Tom Patterson case, using phages to attack and kill a bacterium to try to save a patient's life. But I think we can do better by understanding the biology and understanding the genetics. We can take this further. And so here, antibiotics will likely always be our first stage of treatment in infectious bacterial diseases. They really are a miracle drug. But in antibiotic-resistant infections, I do believe phages can play an important role, and they can really reduce those bacterial loads. But by understanding their biology, we can leverage this further. And we can know and recognize that this will drive the emergence of phage-resistant mutants, which may be resensitized to additional antibiotics, may be more sensitive to the human immune system, and all of this to drive improved clinical treatment outcomes. Now, this is a great idea. Does it actually work? And so this is what we wanted to test. And so we took our phage and antibiotic combination, and we went into a preclinical trial. And so we used this mouse model here, and we had 4 treatment groups. The first group was just a PBS control. We had an antibiotic-only group, we had a phage-only group, and we had a combination of phage plus antibiotic. Mice were inoculated with this bacterial infection, and then 1 hour later, they received that treatment. And then 8 hours later, they received a second dose of the antibiotic only, just due to the short half-life. And this experiment ended at 11 hours. And what we found was that this is bacterial load in the mice, was that with each additional increase in antibiotic agent, we saw a significant reduction in bacterial load along with phage replication. However, we did not see a significant difference between our phage and our combination treatment, which is what we were really trying to show. However, the reason for this was that we had to end this trial early. We had to end this at 11 hours due to the health and ethics of our control and antibiotic-only mice. These mice were incredibly sick with very high bacterial loads, and this simply wasn't long enough for us to select for phage resistance and then to counter this with that antibiotic combination. So we designed a second stage of this trial, just comparing the phage only to the combination treatment. And this trial went for the full 16 hours. And here we're able to show a significant reduction. We also had active phage replication, and we had the emergence of phage resistance, suggesting that yes, this phage plus antibiotic combination is a superior treatment than either on their own. Now, I might jump over this just in the interest of time. This is a little bit more genetics. And I'm going to move a little bit forward here. So what we were able to show here was that phage FGO2 targets the bacterial capsule of Acinetobacter baumannii, and this drives the emergence of these phage-resistant mutants. And we're able to show that this occurs repeatedly, and it occurs repeatedly through these loss-of-function mutations in these capsule-producing genes. And as a result, we have these phage-resistant mutants that no longer have the bacterial capsule. They're less virulent, they cause less severe infections and less severe disease, and they're resensitized to specific beta-lactam antibiotics, and we can leverage this. So we're proposing this as a 1-2 punch combination where we can apply this phage and this antibiotic to improve clinical treatment outcomes. And the point here is that by understanding the biology and the genetics of these phages and how they infect and how they kill their bacterial hosts, we can do much better than what we did in the Tom Patterson case. We can leverage these trade-offs. We can improve clinical outcomes in phage therapy. And so I want to start to take you through what we're doing in this translational pathway. And so this has been an ongoing project that I've been leading over the last 3 to 4 years. This has been in collaboration with Anton Peleg. Anton is a professor in microbiology here at Monash University, and he's also head of infectious diseases at the Alfred Hospital, which is one of our larger infectious disease hospitals in Australia. And what the goal here is to have a local production of phages that we can use to treat Australian or Victorian patients that are suffering these life-threatening superbug infections. And this work is in collaboration with a collective known as Phage Australia, which is really looking to professionalize phage therapy and make this as our 3rd major intervention for infectious diseases behind antibiotics and vaccines. And so I want to go through how we're actually producing these phages to treat patients. And so we've actually established, as Craig mentioned, the Monash Phage Foundry. This is an in-house phage production facility that allows us to produce phages at scale, efficiently, via reproducible methods, and most importantly, phages that are safe. And so how do we do this? Well, the first step in this is that we need the phages, and to get the phages, we need the bacterial host. So we grow up these bacterial pathogens. We do these in large glass flasks, and we shake these at 37 degrees, and we infect these with our phage of interest. And that phage is going to replicate and grow to really, really high numbers. We let that grow for about 16 to 24 hours. And the next day we come in and we run this through a custom-built depth filtration pipeline. So we take this raw bacterial lysate that's got lots of bacterial cells and debris, and we run these through these sequential depth filtrations, which remove the bacterial host. And we then run these through in a sterile biosafety cabinet through a sterilizing grade filter. And we then have a sealed, sterile product that has no bacterial host in it. It just has phages. And these 2 steps are done in our microbiology lab. It's a bit of a dirty research lab, lots of bacteria floating around. But at this point, we've got a sterile product. And so here we move into a new clean room facility, again, based at our Monash campus. And the next step is to wash and remove all of this bacterial byproduct. You can see how yellow this is. This is lots of growth media and other products that we don't want mixed with our phages. And so what we do is we use this machine here. This is a Cytiva Atka Flux 6. We pump our phages up into this large vat at the top here, and then we dilute that 10 to 20-fold with a buffer of our choice. And we run it through this system, which is basically a glorified pump. And we run it through a filter here that's a little bit hard to see. And we cycle this product. We cycle this product under pressure, forward and back pressure, and we concentrate and wash our product. We remove all the bacterial waste, and we concentrate our product down to about 100 mils. And at that point, we have a washed and concentrated product. The next step, which is the most critical, it's also the most time-consuming, and the part that we need more research and investment in, is the removal of bacterial endotoxin. These are products on the bacterial cell wall that cause a very strong inflammatory and immune response in a patient. And so it's critical that we deplete these below safe levels. So currently we're using a mix of these commercial resin kits along with some chemical methods to deplete these endotoxins. This is very painstaking and slow at this point. It can take a couple of days, and really we need some more investment here to get some better equipment to be able to improve this throughput. And finally, we package and produce these phages in single use syringe accessible vials, and we send these off to a third party to have these validated for sterility and endotoxin checks. And so this phage production allows us to treat local patients here in Australia, here in Victoria. And this is enabled through a second initiative, which I co-lead with Anton, known as Vic Phage, which is run at the Alfred Hospital here in Melbourne, and this is the first in-state compassionate use phage therapy service. So how this works, Anton and the clinical team at the Alfred will identify patients suffering from life, limb, or function-threatening infections. These are very severe infections that are antibiotic resistant and have exhausted all other treatment options. When a patient's eligible, we'll isolate the bacterium and they'll be passed over to our labs here at Monash and at the Monash Phage Foundry. We'll either isolate a new phage or use a characterized phage from our collection, and we'll produce that at the Phage Foundry. We'll then pass this back to Anton and team. We'll pass them back a validated, produced batch of phages that they can then administer to a given patient to treat and attempt to treat their superbug infections. And so this, we actually treated our first patient in December 2002. It was the first patient in Victoria treated with personalized phage therapy. This was our first production batch that was used to treat this patient. We produced over 150 vials. Each vial contained a single dose with a set number of phages and a validated level of endotoxin that was safe for intravenous administration. We sent 10% of this batch out for a third-party validation to check both endotoxin and sterility to ensure our products were safe. And then we passed this batch over to the clinical team at the Alfred. Now, this patient that we treated was a 21-year-old cystic fibrosis patient who was suffering a really, really horrible bacterial infection known as Bordetella bronchialis, very, very drug resistant, and it was causing consistent bloodstream infections in that patient. It's a very complex case, but it was really exciting and rewarding to go through this whole process and actually begin to treat this patient. And we were actually followed by a documentary team throughout this whole process, which really added to both the excitement, but also the, I guess, a little bit the terror at doing all of this. And so this documentary came out a couple months ago. You can find it on SBS On Demand. It's called Last Chance to Save a Life. Obviously, I'm very biased, but it's an incredible story, and it really tells a personalized story. It tells the patient, follows the patient, their family, how much these antibiotic-resistant infections really impact people's lives and everyone around them. It then follows the medical team at the Alfred going through the treatment. It comes to our labs and follows us as we isolate, as we produce the phages and the time pressures that we had during that. And finally, we see the administration of phages to these patients, which was the first time that it happened in Victoria. So really exciting to see this out. And if you're interested, I'd encourage you all to have a look at that. So with that, I'm going to wrap up. I'm going to give a big thanks to my lab, particularly to Fernando, whose PhD work that I presented on, to all of our collaborators, the VicPhage initiatives, particularly Anton and the entire team at the Alfred, Phage Australia, the Monash Bioinformatics platform, all of our funders. And thank you all for listening. Really hope you enjoyed it, and I'm more than happy to take any questions. So thank you.
Thanks so much, Jeremy. We do have some questions coming through. So I'll ask those on behalf of the people who've asked them. So first up is, what are the potential dangers of phage therapy? How safe is it, and is there any risk to the patients that you're treating?
Yeah, so it's a great question. Look, broadly, I will say phage therapy is safe. It's actually a lot safer than antibiotics in some respects. Antibiotics can have a lot of side effects. They can affect your gut. They can cause immune responses. They can have a lot of off-target effects in affecting different organs. In that direct comparison, phages are really safe. Phages are actually present in your body. They're in your gut. They're circulating in your body already. So a phage itself is very, very safe. However, the difficulty is in producing these. And because we have to grow these in these antibiotic-resistant bacterium, and those bacterium, those products can be quite dangerous. And so the risk is really in producing those phages at a safe level. And that's why we make sure we've got all these checks and these quality controls that the phage products that we're producing are at a safe level that we can introduce to a patient. So still a lot more data we need to generate here, but in general, it's been quite a safe and effective treatment.
Great, thank you. And I've got quite a nice follow-up that follows on from the production. So given that the screening process that you talked about, it seems quite manual and laborious. So the specific question is there a chance to steal some of the robotics from crystallographers or someone else to increase the throughput of that screening process?
It's a great question. And I will firstly say that it is quite manual and it is quite laborious. It takes us about about 2 weeks to produce a phage and have that validated and ready. We can do it faster. I would prefer not to do it faster because it's quite stressful. You know, 2 weeks from some patients' infections, if you've got a really severe bacterial infection, is not fast enough. You know, some of those you might need responses in hours to days. So there's a lot of work that we need to do to improve this and increase that throughput. And you know, the robotics point is an excellent idea. There are some commercial groups and some large research labs that are using high throughput robotics to really speed up the phage screening process, which is one of the more time-consuming steps, and try to get a result and a phage that can infect and kill a patient's bacterium in a matter of hours. And that would really speed up the process. And I think automating some or getting semi-automated equipment for some of our steps would really reduce our time footprint and allow us to treat more patients.
So given that they do have to be sort of produced individually for each case, do you see this becoming widespread?
Yeah, it's a real challenge at the moment. So in my opinion, I think phage therapy has lots of potential use cases. But right now, the approach that I sort of went through there is very personalized. And realistically, you know, our team, I think we could look at treating between 5 to 10 patients a year at the current scale and investment that we have. And you know, that's incredible. For these patients, they may not have any other treatment options. It may literally be their last chance. And so that's incredibly rewarding. But if we look at the antibiotic resistance crisis, this is continuing to grow. It's really going to be a slow pandemic. It's predicted to become one of the top killers in the next 10 to 20 years. We do need to speed this up. And so there's a lot of ways that we can do this. We can produce what we call cocktails, so combinations of phages that may work against larger groups of pathogens. And some of the new work that we're doing is actually trying to build specific products against pathogens at the Alfred Hospital and have these ready to go so that we can speed up the processing time and treat more patients. But it's a big challenge at the moment.
So given that limited capacity, sort of 5 patients a year, how long does it take to diagnose whether a particular patient would benefit from this, whether they've got the sort of infection that needs this kind of treatment? And how long is it from identifying that patient for them getting treated and recovering, perhaps?
Another great question. So it really varies is the answer. So at the moment, we've treated 3 patients with this approach. All of those patients have had chronic infections. So these have been infections that they've had for years, which is really horrible for these patients and their families. But from our perspective, they're a much slower infection. And so that gives us time to produce these therapeutics, to produce these phages and have a treatment impact on those patients. We do want to get to a point where we can respond to some of these more acute infections, so bloodstream or soft tissue infections. That's going to require us to be prepared and ready and have phages on hand that are ready to go against some of those patients. So in terms of how the hospital staff identify these, they have to fit these regulatory guidelines, meaning that they have to be life, limb, or function threatening. So it has to be a really severe infection, exhausted standard of care, so there are no antibiotics available. And that's really a clinical decision. So Anton and his team decide who is eligible for these treatments.
So given what we know about the array of multi-drug resistant bacteria, is it possible to foundry these phages and store for over long periods particular cocktails that could be used for drug-resistant bacteria that we know of?
Yes, absolutely. And we're doing some of that work here. So some of our more recent work, we're actually targeting a nosocomial pathogen. So this means that this pathogen is present in the hospital. It's present in the hospital at the Alfred. It has a reservoir somewhere in the hospital, likely in the sinks and the drainage. And patients that come into that hospital for surgeries or other reasons are picking up these infections in the hospital. And so it's a really horrible situation. And these are very virulent and also very drug resistant. And so what we're doing is we're producing these phages and we're pre-targeting them to make sure that they can be effective against very specific pathogen groups. So it is possible, it is feasible. The limitation is that phages are very, very specific. And so we are not going to be able to have a phage therapy that's going to work against every bacterium. And so instead we need to we need to produce collections of phages, cocktails of phages that will be effective against a group of pathogens. And it's likely we're going to need many, many, many of these products to cover the array and diversity of infections that we're seeing.
And is isolation the only way to get the phages, or is there synthetic methods that you could use to create artificial phages that could be done sped up quite rapidly, or spun up quite rapidly?
That's an excellent question. The short answer is yes, there are synthetic methods, but they're very early stages. So, one of the amazing things about phages, they're in some respects a very simple organism. They're a small genome, a protein structure. We know a lot about them. And so they've actually been used as key tools in a lot of molecular biology, a lot of sequencing. And so we know a lot about them, and they've led to a lot of discoveries. So currently, yes, we're going out and we're isolating phages from sewages, and we're characterizing and we're producing them. I have no doubt at the end of my career, we will be printing phages. We'll be rebooting these through synthetic methods. We'll be designing their genomes through informatics and utilizing AI, and we'll be producing targeted therapeutics. And I hope to be able to tell my graduate students when we get to that stage that I used to isolate phages from the environment, and they'll laugh at us and think that that is ridiculous. When we do that. So we will get there. It's going to require a lot of research, a lot of investment in understanding this. But yes, there are ways that we can synthetically produce these.
And would this synthetic production provide an avenue to perhaps attack some where it's difficult? Are there particular groups of bacteria where it's difficult to isolate phages that'll work for them, and maybe we could design phages that will fit that kind of criteria?
Exactly. That's kind of the hope and dream that, we could take large sequencing data sets. So we go out in the environment, sequence and find all of the viruses and the phages in that environment. And then we could isolate the genes, and then we can make these modular. We could take a tail from this phage, a replicative cassette from another phage, and recombine them synthetically to produce them to make a very targeted antimicrobial against hopefully any pathogen that we desire.
Just a couple more questions are coming through. So you talked about the need to remove those endotoxins in order to ensure that what you deliver to the patients is safe. What makes that a particularly challenging step?
Yes, a lot of reasons. So endotoxins they're very hydrophilic, they're negatively charged, and they tend to aggregate right around the same size as a phage. So a lot of our methods use size selection, where we're using filters, and different pressure and biophysical methods to separate out the phages from other products. And the endotoxins really co-aggregate with those phage products. And so what we really need to use are sort of chemical methods and chromatography methods to selectively remove the endotoxins. We've got some very, very good methods that remove the endotoxins, but they unfortunately also remove the phages. So it's finding that balance of what's selective and what's going to work in a reasonable time frame.
And after you administer the phages, do you see evolution of the phages happening in patients such that the bacteria and the phage are in a kind of arms race trying to escape and attack one another?
We absolutely do. We've got a patient we're treating right now at the moment at the Alfred, another cystic fibrosis patient, and we're seeing some of that in real time. So this patient's now going through their second administration of phage therapy. And we're seeing that the original bacterial pathogen that we treated is shifting and changing, and we need to shift and change our phages with it. And once you administer the phages, they're in that patient. They're going to continually infect and replicate and persist with that bacterial pathogen. So it's really interesting. It's not a lot known about that space at the moment, but we've got a number of research projects sort of following up in that area.
Cool. And last off, I'm going to go with something of my own. So you mentioned at the beginning that phages are incredibly diverse and abundant. As you know, I work on insects, and one of the things that I say about insects is that they're incredibly diverse and abundant. So I wonder how many phages there are, and how many do you think you'll actually need to combat the array of multi-drug resistant bacteria that we see?
Yeah, so it's a really cool thought experiment to try to extrapolate how many phages are on the planet. And thankfully, some other brilliant scientists have done this. And so the numbers are an estimated 10 to the power of 31 phages on the planet. So 31 zeros after that, which is a number that you can't even comprehend. So there's more phages than grains of sand on the planet. There's more phages than observable stars in the universe. They are the most abundant on the planet, which gives us a lot of hope that we've got this huge, really almost unlimited resource of these viruses that are going to continually adapt and evolve. And so the good thing is that we'll never run out of phage. So we are running out of antibiotics. That will never happen with phages because they're biological. They evolve and change with their bacterial host. But it also brings a lot of challenges. Finding the right phage, finding a phage that works and administering that to a patient in a effective time frame are some of the challenges we're facing right now.
Yeah, wonderful. Thanks so much, Jeremy. And I'll just pass on this. Also, a large number of thank yous have come through and congratulating you on a wonderful talk.
Yes. Look, thank you. Congratulations again, Jeremy, for a wonderful talk. I think we certainly can see lots of questions that have come through. It's certainly been informative, lots of things to think, and quite important work as well. So thanks for taking the time. And thank you, Craig, as well, for taking the time to moderate the session. To all our alumni, guests, and students that have joined in today, look, a huge thank you to you. Certainly, join us for our webinar series that will be happening every couple of months.